All right, so this lecture video is going to cover genetics and heredity. I feel like that's really dark, but I don't have great lighting in my room right now. So we're going to talk about Gregor Mendel. You should have learned about him in 7th grade as well. So he is known as kind of being the father of genetics. He's the one that discovered the laws of heredity that we know is our most basic information about how traits are passed from parents to offspring. When he worked with pea plants in the monastery that he lived in, he was a monk. He's Austrian. Mid-1800s, as you can see there, and he was in charge of the garden, and so he did a lot of breeding of pea plants in order and took copious notes to determine the most basic information about the laws of heredity. So we've already defined heredity at the beginning of the year. It's the passing of traits or characteristics from parents to offspring. Genetics is specifically the branch of biology that studies heredity. We've covered traits, genes, and alleles. Not alleles quite so much, but genes specifically a lot lately. So traits, these are inherited characteristics that are going to vary from one individual to another. So for example, in humans, eye color, okay? Genes, these, as we've just recently been talking quite a bit about, they're segments of DNA that code for a specific protein and then therefore determine a specific trait. And every diploid cell in organisms is going to have 2 alleles, right, that are the different gene forms that are going to code for the makeup, genetic makeup overall, the genotype and the phenotype. We'll get into that soon, of a particular organism. Okay, so if we go back to that for humans, to just keep this simple, we have 23 different types of chromosomes. We get 2 of each type from our parents, 23 from the female and the egg cell, and 23 in the sperm cell, and then those are going to fuse to give us a total of 46 in the diploid zygote that can grow and divide by mitosis to make a multicellular organism. Okay, so all of that being said, every single one of the genes that we're going to talk about, right, has these 2 alleles. Like in my cells, I have an allele from my mom and an allele from my dad for every trait. Right? These are always going to be located at a specific locus on a specific chromosome. Right? Hang on just a second. All right, so this is going to kind of hit on some of the historical context of Mendel so that we can understand what he discovered essentially working with plants, since a lot of times we just don't think about the fact that plants are going to reproduce sexually. So he took pollen from one plant and transferred it to another plant, okay? Or he took pollen and he transferred pollen from a single plant to the same single plant, okay, depending on what he was doing. So he kind of started out working on just true breeding situations where he allowed the plants to self-pollinate, or he, I should say, moved the pollen from plant A from the male part of plant A to the female part of plant A, okay? Pollen contains sperm cells. Right, and then within the ovary of a pea plant, because they produce flowers, there's going to be an egg cell. Okay, so he just moved pollen from the male to the female part of the plant on the same plant. Okay, so that they're going to have the same exact traits as their parents for sure. Then he did that a lot with all these different traits that you can see in this table. Okay, so then he crossed 2 different types of plants that again we can see in the table. Okay. So that there was a combination of traits from 2 different parents. This vocabulary is just some basic genetics vocabulary that you're going to see in working problems. So the parental generation is the P generation. The offspring of that cross from the 2 parental organisms, that's going to be the F1 generation, which stands for first phylele. And then if you were then to take offspring from the first phylele generation, you get the second phylele generation. Kind of think of this like a family tree, okay? Your grandparents, then your parents, then you. Your grandparents would be like the parental generation. Your parents would be the F1. You would be the F2, right? If you then way down the road when you meet somebody nice and you fall in love and you get married and you have kids, then they would be the F3 generation if we started with your grandparents, okay? Just like I said, a little bit of context there. Okay, so that being said, if we go to Mendel's studies with plants where he dealt with producing offspring that were hybrids, okay, that's what we're seeing here in the table. So these were the traits that he studied, seed shape, seed color, okay, seed coat, etc. He crossed this parent that we see along the top, round all the way to the right with tall, versus, okay, wrinkled all the way to the right with short, okay, and he focused on this trait, and then another time he focused on this one, another time he focused on this one, and he produced hundreds and hundreds and hundreds of offspring plants as a result of these crosses that he did, okay, and he basically always got the same outcome mathematically, right? So when he did these two crosses initially, he always got this as the offspring, okay, that we see going left to right there. Okay, so what that led him to was his first principle, right? And that's the principle of dominance. Okay, so just real simple. Some alleles are dominant and some are recessive, right? So what does that mean? Okay, we've already talked about this a little bit too. And we're going to use capital letters for dominant alleles. Recessive alleles are going to be written as lowercase letters. You can choose any letter that you would like to use, but you need to use letters that are definitely different in the capital and the lowercase form, like a T, like an A, like a D, okay? So dominant alleles, these are alleles that are visible and observable, okay, or expressed. If they are present with the recessive allele, they're going to mask that recessive allele or cover it up, okay, when they're both present together. Recessive alleles, these are the alleles that are going to be covered up or masked by that dominant allele if they're both present together. Okay, so here's an example. In Mendel's plants, if they had the offspring had two alleles for being tall, that's how we're going to write that, two capital T's. Two alleles for being short, one from each parent, one from the egg and one from the sperm. We're going to write that as two lowercase t's. And then if they have one of each, one dominant allele from, let's say, the egg cell, and the other one is recessive and it's from the sperm cell, then we're going to write it like this, one capital and one lowercase letter. And so then therefore, that capital T is going to mask that recessive T, right? Well, what does that mean? All right, so let's just kind of start. That's what we're doing with those heterozygous individuals. A pure breeding tall plant crossed with a pure breeding or true breeding short plant. We're going to get tall offspring. All of them are going to be tall, but they're going to have one allele that's for tall and one allele that's for short, okay, in this particular generation. All right. So then, after Mendel did that, and he kept getting the same outcome every single time, then he decided he was going to cross two individuals from this F1 generation together. And that's essentially, when he looked at the outcome, that's how he came up with his second principle, okay? So his second principle is called the principle of segregation, right? And so this is that the alleles are going to segregate or separate as cells divide, right? So this says this occurs when the chromatids separate during anaphase 2 of meiosis, so that each gamete is going to have only one allele for a given gene, right? So remember, homologous pairs are going to separate during anaphase 1, and then anaphase 2, the replicated chromosomes have to split up, right? So that in meiosis, each new cell gets half of the original amount of DNA, right? And then, so basically, here's his cross, okay? These two plants are both heterozygous, right? And so then when he did this a whole lot, he got, most of the time he got tall plants, and every so often he got short plants, okay? And so this is how, again, getting these short plants occasionally is how he came up with this second principle, okay? So then let's move to the next slide, right? And one more thing too, randomly parents are going to pass on certain egg cells, certain sperm cells, okay, as far as just the genetic makeup, and that's kind of what's mentioned right there. Okay, so we're randomly going to get one allele from each parent, depending on what the gamete that is used is. Okay, remember gametes are either egg or sperm. All right, so next, let's talk about probability. Okay, so probability, just by definition, is the likelihood that a particular event is going to occur. So when we talk about genetic outcomes and what's possible in the offspring, we are basing this on probability, and your outcomes are going to be pretty consistent, okay? When we're working with a Punnett square that has 4 boxes in it or is a monohybrid cross, there's not a lot of options for outcomes when we're following Mendel's rules, okay? So your basic equation for probability is going to be the number of one kind of possible outcome over your total number of possible outcomes. And we're going to always deal with a total of 4 to get started here when we're working monohybrid Punnett square problems, okay? One thing to mention, past outcomes do not affect future outcomes. right? The alleles are going to segregate randomly every time gametes are formed, okay? So here are some examples. Every time you flip a coin, you got a 50-50 shot of getting heads or tails, right? Every time you roll a dice, right, because they have 6 sides, you have a 1 out of 6 chance of getting a 4 or getting a 3, whatever it happens to be, okay? Let's say you roll a dice 5 times. You could get a 3 all 5 of those times, right? If you roll it 5 times, the first roll has no impact on the last roll, right? And vice versa, okay? Then sex of a child, 50-50 shot every time that a baby is born, okay? 2 of those sperm cells are going to contain a Y chromosome. 2 of the sperm cells are going to contain an X chromosome. More to come on that eventually, right? So you can't, even if there's a family that says they wish that you say they have 3 girls and they want to have a 4th baby, and they say, oh, hope we have a boy, they still could get a 4th girl, okay? All right, so then Punnett squares are going to use probability for us as students, teachers, scientists, whatever, to find expected ratios when genetic crosses take place, okay? So notice that this says with this little bullet, these do not represent the actual outcome, like I just said, okay, as far as male versus female children, that in theory, somebody could have a family that, you know, they've got 7 siblings, so there's 8 kids and they're all males, okay, or they're all females, right? So this is just what's possible, okay? So and then when we look at more individuals in a sample, the closer you get to those expected ratios, and that's basically how Mendel figured this out, is that he bred his pea plants over and over and over again, and he kept getting the same exact ratios every time, right? Which is what led him to say, okay, this has got to be how this is working, okay? So, and I realize with a picture and a Punnett square, that's not typically what you would do, right? But that's what we're seeing here. So we have got these 2 plants of Mendel's. I'm going to refocus for just a second, see if that gets a little more clear. These 2 plants of Mendel's that are both heterozygous, tall plants, and I'm going to rewrite that because I feel like the alleles look really blurry. Okay, and so again, this would technically be the male parent, and then this is the I don't think I've said this yet. And then the female parent on the side, okay, male typically goes on the top, and female on the side if you know which parent is which. Okay, in this case, we don't. All right, but if it says a man and a woman, man's alleles would go up here, woman's alleles over here. All right, and so all of that being said, okay, these alleles are going to separate during anaphase 1 of meiosis. That's on another slide that we just talked about, okay? And so this, with me making those 2 little diagonal lines, okay, that's kind of hard to see. Let me do it over here. If I write this genotype of that parent like this, and then I do this, that's showing anaphase 1 and that homologous pair separating. So then you write the parent alleles, okay, over the boxes or to the left of the boxes. You draw your Punnett square, okay, and then you're going to fill that in, all right? Let me see, hang on. So then each gamete, okay, the eggs or the sperm, they're going to have 1 allele for every trait at the end of meiosis, okay? And so then that's your parent alleles on the outside. These are your possible offspring on the inside, okay? So that then when we write ratios, right, that we are going to see the same outcome every time. All right, so there's that particular Punnett square. Maybe a little bit clearer without having the plants in there as well, okay? So then the next slide, let me come back down here. We are doing what's called a monohybrid cross when we're drawing Punnett squares that have 4 boxes and looking at a single trait and the alleles that are passed from parents to offspring. So your total, your 100%, okay, is always going to be just 4, right? And so then how do we interpret this, okay? So let me go back to this, right? That's the problem that we've just seen in the example, okay? So the allele combinations in the offspring are on the inside, okay? So those allele combinations are called genotypes, and then the physical expression is called the phenotype, okay? So phenotype, physical, right? So that's tall versus short, okay? Then the genotype is the allele combination of an organism that we're going to get one allele from the female parent, one allele from the male parent, okay? So then if the genotype of a tall plant with 2 tall alleles is homozygous dominant, and that's how we would read that, okay? Then a genotype of a tall pea plant with 1 tall and 1 recessive, or sorry, 1 tall allele and 1 short allele, all right, then this is going to be heterozygous, okay? And then a genotype for a short pea plant is going to be homozygous recessive, right? And that's how you read those particular genotypes, okay? You don't say like big T, or little t, little t. You don't sound very smart when you say that. You say homozygous dominant, again here, heterozygous or homozygous recessive. Like if I was working a problem on the board and I was asking you what I needed to write, you would use those words instead of big T, big T. All right, so then here are those vocabulary words that I've just been using, okay? So homozygous organisms can be homozygous dominant or homozygous recessive. These are the ones that are pure or true breeding. Okay, heterozygous individuals are going to have 1 dominant and 1 recessive allele. Okay, so these are hybrids. Don't use that term too much, or the pure or the true breeding. Okay, so if an individual is heterozygous, it is carrying a recessive allele. That recessive allele is masked or hidden phenotypically underneath, behind, however you want to think about that, behind that dominant allele. Okay, the term carrier is going to be more important with when we talk about genetic disorders and some things like that. It's not super important to know that a tall pea plant carries a recessive allele because it's heterozygous, but for certain characteristics it's going to be real important to know that, okay? And so then at the end of completing Punnett squares, then you're going to write what are called genotypic and phenotypic ratios of the offspring. All right, so I'm going to stop there with this one, and I'm going to say this lecture needs to come before the one that says solving genetics problems, where I work through how to solve them on that particular sheet that's part of your notes. It says that solving genetics problems at the top, okay? And then there's one other video posted that's just me working through the 2 bottoms at the bottom of that sheet. But make sure that you watch both of those after this particular lecture. Let me know if you have any questions.